Good morning, everyone. So today we will discuss about DNA. So far, you have learned about cell ultrastructure and chromosomal structure and morphology and the chemical composition and all those things. And last class we discussed about chromosomal theory of inheritance. That means the genes are located on chromosome. So today we will discuss about something different topic. The first slide says that. So just I wanted to ask everyone a question about what is the secret of life. So there are so many life forms on this earth, from virus to so evolved animal called a human being. So what is the secret behind all these living forms on this earth? So is it a cell? So for this mystery or for this unanswered question, so on February 28, 1953, two people, like guys, walked into an Eagle pub in the Cambridge, that is in United Kingdom presently. So Eagle pub, they said that they have an answer for the secret of life. So this is the Eagle pub. So pub means this is the place where the scientists used to go there and they used to drink and think about the science. So two guys, that is Watson and Crick, so they walked into this Eagle pub, so which is in the United Kingdom, that is in Cavendish Laboratory, Cambridge University, and they said that they have an answer for life, that is secret of life, that is a DNA. So this is the double helical structure of DNA. They told that DNA is the secret of life uh, of all the organisms. So today we will discuss about DNA, that is deoxyribonucleic acid, different types of DNA, and structure of DNA, and replication, and what is the function of the DNA. So this is the overall snapshot of today's topic on DNA. So these are the metaphase chromosomes which are duplicated in the S phase, and if you go further into this chromosomes, these are called the scaphoids, so looped DNA. And if you just zoom into these structures, condensed scaffolds, so these are the loops. So if you go to this further, if you go to these loops, so here a 30 nanometer fiber DNA is there, which is wrapped in um, histone proteins. So if you go still further about this 30 nanometer fiber DNA, so that is a 10 nanometer fiber. So that means these are the beads and the string. So this is like a string and these are like a beads. So these are called nucleosomes. So if you take this part and highlight this part, what it is, this is the naked DNA, which is double stranded DNA. So this is the overall picture of DNA from its chromosome. So this is the roadmap. And so one can say so how the DNA is condensed during um, meiosis and mitosis. So who helped to discover this double helical structure? So none other than Rosalind Franklin. She was working with Wilkins. So one more guy, in addition to Francis and Crick, Rosalind Franklin was working with Wilkins. So Rosalind Franklin, she was a chemist and she was a X-ray crystallographer. She was she captured the picture for Wilkins. So this is the photo 51. The story behind double helical structure is this, the picture. So photo 51, this is the X-ray crystallography which was taken by Rosalind Franklin for Wilkins. So this is how she did an experiment. She crystallized the DNA and she passed the X-rays through this crystallized DNA. So then these rays were captured on a photographic film. So that is what is this picture is. So this shows the double helical structure of DNA. So this is by X-ray diffraction study conducted by Rosalind Franklin for Wilkins. But based on this, the Francis Crick and Jim Watson in 1953, they told that the DNA is has double helical structure, and which has 4 nucleotides, that is adenine, thymine, guanine, and cytosine. So this was the paper they published in Nature journal, Genetic Implications of the Structure of Deoxyribonucleic Acid by J.D. Watson and F.S.C. Crick. So this was the original paper. So this was the original picture they draw when they published this paper in 1954. So in 1962, Watson, Crick, and Wilkins got a Nobel Prize. These 3 people got Nobel Prize. This is Watson and Watson, Crick, and Wilkins. These 3 people got Nobel Prize in 1962. So why this Nobel Prize was not shared with Rosalind Franklin? So because she died in 1958, whereas they got this Nobel Prize in 1962. So it was not awarded posthumously, means after death it was not awarded to Rosalind Franklin. So that's why she is known as feminist icon. She is the flawed perfectionist. So this is the book written by Brenda Maddox. So Rosalind Franklin, The Dark Lady of DNA. So if you get a chance to read this book, read it. It is very good book. So I have read this book also. So about the secret of photo 51, there is a video which is available in this website. Whenever you get time, so you can watch this video also. And about, I have one more video. I have that video, but I cannot show. Whenever we get a time in college, I may invite everyone to watch this short movie made on secret of life. So this is also, if you want, I can share the link. You can watch. So what is the secret behind DNA discovery? So with this, we will see what are all the historical milestones of this discovery of DNA. In 1869, Friedrich Mischer, he was the scientist who discovered or first isolated nucleic acids from first cells and named them as a nuclein. So in 1884, one more scientist called Hertwig, so he proposed that nuclein to be the carrier of hereditary traits. So in 1899, Altman is told that nucleic acids are acidic in nature, so they were named as nucleic acids and then finally named as nucleic acids. So in 1880, Fischer discovered the presence of purines and pyrimidine bases in the nucleic acids. In 1910, scientists by name Levin found DNA contains phosphoric acid and deoxyribose sugar. So you know that in addition to nucleotides, this DNA contains phosphoric acid and ribose sugar. So he characterized these 4 types of nucleotides present in DNA, that is adenine, thymine, guanine, and cytosine. In 1950, one more scientist by name Chargaff, he told that the content of purine and pyrimidine in DNA is equal. And that means purine to purine, like guanine and adenine, equal proportion, and cytosine and thymine are in equal proportion. So we'll study what is Chargaff's rule in the future slides. In 1950, W.T. Asbury found through X-ray diffraction that DNA is polynucleotide with nucleotides arranged perpendicular to the long axis of the molecule and separated from one another by a distance of 0.3 nanometer. So see, he told that through X-ray diffraction, so DNA is just like a polynucleotide. Polynucleotide means number of many number of nucleotides which are arranged in a particular a perpendicular fashion to the long axis of the molecule. In 1953, Wilkins and Franklin got a very fine X-ray photograph of DNA, and they worked out that first correct double helix model from the X-ray photographs of Wilkins and Franklin. Franklin means Rosalind Franklin, and Wilkins. These two people were working in the same laboratory. And in 1959, Kornberg, so he did an in vitro synthesis of DNA, that is artificial synthesis of DNA. And in 1962, Wilkins, Watson, and Crick, these three people were awarded a Nobel Prize for the discovery of double helical structure of DNA. So now we will discuss about DNA in detail. So DNA is the genetic material found in almost all the cells of all living organisms, and DNA is the fundamental building blocks for life. So I told, so all living forms on this earth have just 4 letters, A, T, G, C. So how these different lives have been evolved means the arrangement of these nucleotides. So if you take any life, they have only just 4 letters. The way these ATGC have been arranged in these organisms, so made, they are very unique on this earth. So most of the DNA is located in the nucleus of the cell. So it is called as nuclear DNA. And if the DNA is also found in mitochondria, it is called as mitochondrial DNA, which is circular in nature, which we have already discussed when we were discussing about cell ultrastructure. And some amount of DNA is also found in chloroplast, especially in plants. So it is called as chloroplast DNA. Again, this chloroplast DNA is circular in nature. So next, what is the composition of these nucleic acids? So nucleic acids means it is both DNA as well as RNA. So the main composition of these nucleotides or nucleic acids is phosphoric acid. So this one, the golden color ball, looks like the golden color ball. This is the phosphoric acid or phosphate molecule, which is there in the polynucleotide chain of this DNA. And the second composition of this nucleic acid is pentose sugar. So this is the pentose sugar, which has 5 carbon molecules. So this is the pentose sugar. And the next one is organic bases. So these are the organic bases. So in case of DNA, so we have adenine, thymine, guanine, and cytosine. And in case of RNA, so along with this phosphoric acid and pentose sugar, so we have like adenine, uracil, guanine, and cytosine. So just observe this structure, the ultrastructure, molecular structure of this DNA. So there is a direction shown here with the arrow. So this is the 5 prime 3 prime direction, and this is 5 prime to 3 prime. So we always know that whenever we tell these nucleotides, we always tell in this fashion, 5 prime to 3 prime, not 3 prime to 5 prime. So why it is so, I will tell in the future slides. So this is the structure, backbone structure of DNA. So it has phosphoric acid, pentose sugar, and the organic bases. So these organic bases are paired. So this guanine and cytosine have paired with 3 hydrogen bonds, and thymine and adenine paired with 2 hydrogen bonds. And first we will discuss about how this phosphoric acid looks in the polynucleotide chain of DNA. So phosphoric acid, it is the chemical, it is H3PO4, has 3 reactive oxygen groups. So it has 3 reactive oxygen groups, these 3. So these 3 are the reactive OH groups, of which 2 are involved in forming sugar-phosphate backbone of DNA. So out of 3 OH groups, reactive OH groups, so 2 are involved. So this one and this one are involved in formation of sugar-phosphate backbone of DNA. So where exactly these sugars and this phosphoric acid are phosphate molecules bind means, so this is the first one. This OH group of the phosphoric acid binds to 5' carbon atom of the sugar. So this is the sugar, this is the 5' carbon atom of the sugar. So one OH group binds to 5' carbon atom of the sugar, and another binds to 3' carbon atom of the sugar. So that means, so out of 3 active reactive OH groups, 2 are involved in sugar and phosphate backbone formation of the DNA. So similarly, in case of a DNA, the same thing. 2 molecules of OH groups are involved in 5', 3' sugar phosphate backbone formation in DNA. So this is about RNA. So what is the structure behind this? And this is the DNA nucleotide structure. The only difference between RNA and DNA is, so I'll come to that point in the next slide. So this is the summary of this slide. So how this phosphoric acid is involved in sugar phosphate backbone of a DNA. 5' carbon atom will bind to 5' carbon atom of the sugar, and 3' carbon atom of the sugar will bound to this phosphoric acid, which forms the phosphodiester linkage bond. So nomenclature. So the number of phosphate groups determines the nomenclature of these nucleotides. So if this nucleotide, like any nucleotide, so CH2 group, so any nucleotide in this site may bind to one molecule of a phosphate group. It is called as monophosphate. Means a base which is bound to only one phosphate group is called as monophosphate. That is an example for adenine. We can say adenosine monophosphate, AMP. So TMP, GMP, and CMP. And if the base is bound to 2 carbon atoms, it is called diphosphate. And if it is bound to 3 phosphate groups, it is called as triphosphate. So ATP, so adenosine triphosphate. So you heard of this ATP is a energy, so a storing unit. So I wanted to ask a question. So among this AMP, ADP, and ATP, so though the nucleotide is same, that is adenine, but it has different molecules of phosphate. But highest energy is stored in ATP, not in ADP or not in AMP. So why is it so? So you can message me, or you can call me or tell me why the highest level of energy or more energy is stored in ATP rather than ADP or AMP. So this is how the adenosine triphosphate looks. This is the sugar molecule, and this is the adenine, which binds to first carbon atom of the sugar, and the phosphate, the second molecule of phosphate, and the third molecule of phosphate. So this forms an ATP, adenosine triphosphate. So base, sugar, and 3 molecules of phosphate. So now we will study the second composition of this DNA or nucleic acid. DNA and RNA is a pentose sugar. So this is the sugar molecule present in DNA. This is called as D2', means 2' deoxyribose, and the sugar which is present in the RNA is called as ribose. Just observe this 2' carbon atom of this sugar, ribose sugar, and the 2' deoxyribose sugar. So there is a absence of OH group in the 2' carbon atom in the deoxyribose sugar, that is in the DNA. So as compared to the presence of OH group in the ribose sugar. So this is the only difference of this pentose sugar which is present in the DNA and the RNA. So RNA has a ribose sugar which has a 2' OH group presence, whereas DNA has 2 deoxy. Deoxy means the absence of OH group in the 2' carbon atom. So this is the main difference. So that's why it is called as deoxyribo sugar, and this is called as just a ribose sugar because it is having OH group. So and the 5', 3' carbons of the pentose is involved in phosphodiester linkage. So this 3' carbon atom and the 5' carbon atom, this one. So if you name this carbon atom 1, 2, 3, 4 and 5, the 3' carbon atom and the 5' carbon atom are involved in phosphodiester linkage formation. So this is where the phosphate groups will bind 3' and 5'. So, and what about the first carbon atom of the sugar? So to which this first carbon atom of the ribose sugar or deoxyribose sugar, the organic base or the nucleotides will bind, like whether it is A, T, G, C, or in case of like RNA, it could be A, U, G, C. So I hope you are clear about the pentose sugar which is present in DNA and the RNA. The only difference is the 2' carbon atom has OH group in case of RNA, whereas OH group is missing in the DNA backbone, that is pentose sugar. So now we will see nitrogen bases. So what are all the nitrogen bases present in DNA and RNA? So these are grouped into pyrimidines and the purines. So in case of pyrimidines, so these are the just single ring structure. Single ring means it is a benzene ring. Single ring structures. So there are 3 nitrogen bases comes under pyrimidine category, which is called as cytosine, thymine, and the uracil. So if you want to just remember, just use this notation as PyCUT. PYCUT. Pyrimidine. Py means pyrimidine. C means cytosine. U means uracil. T means thymine. PyCUT. And the second category is purine molecules, in which there are 2 molecules, guanine and adenine. So these are double ring structures. Just look at these structures. So and one more thing is I wanted to tell is nobody will ask these structures in the exam. So don't worry. But you should know whether it is single ring structure or the double ring structure, and what are all the purine molecules and what are all the pyrimidine molecules. That's what you should know. So nobody will ask the structure, but if you write this structure, it is wonderful. So these purines can be remembered by simple acronym PUGA. Pu means purine, G means guanine, and A means adenine. So these things are not available in the textbook. It is not a universal code. So just I have given, just to remember. So in case of DNA, so out of these 5 nitrogen bases, so A, T, G, C are present in DNA, whereas AUGC is present in RNA. That means a thymine molecule is absent in RNA, whereas uracil molecule is absent in DNA. This is very, very important. A, G, C are common, whereas T and U are unique to DNA and RNA respectively. So now, how do you name these nucleotides at the backbone? So one terminology I wanted to tell is nucleosides. So nucleosides means if the base is bound to sugar is called as nucleosides. Yes, sides, base plus sugar. If the linkage between the ribose sugar and the organic bases means nucleotides, in case of RNA, will yield ribonucleosides. So whereas if the linkage between deoxyribose in DNA and any organic base will yield deoxyribosides. Just remember ribonucleosides in RNA and deoxyribosides in DNA. So this is how, this is the sugar and this is the base. So base plus sugar, it gives the nucleoside. So these are the ribosides in RNA. So based on the base and the sugar composition, if it is in here in the base position, if there is adenine, it is called adenosine. If it is guanine, it's guanosine. Uridine, uridine. Uracil, uridine. And cytosine is cytidine. So in case of DNA, if it is adenosine, it is called as deoxyadenosine. Guanine means deoxyguanosine, and cytosine means deoxycytidine, and thymidine means if the thymine is present in place of this, in place of base. So nucleotides, the second nomenclature is nucleotides. So we discuss about the nucleosides. Base plus sugar is nucleoside, whereas base plus sugar plus phosphate group is a nucleotide. So nucleotide is formed when a phosphate group is attached to either 3' or 5' of the molecule is known as nucleo 3' 5' carbon molecule of the nucleoside is called as nucleotides. So based on this, we can categorize as ribonucleotides in case of RNA, that is 5' adenylate acid, 5' guanylic acid, 5' cytidylate acid, and 5' uridylic acid. So whereas in case of DNA, it is known as deoxyribonucleotides. It is deoxyadenylic acid, deoxyguanylic acid, deoxycytidylate acid, and thymidylate acid. So this is the structure. So how the nucleotide looks. So this is the sugar, and this is the adenine base. Example I have taken adenine base, and which is attached to the phosphate group. This is the overall structure of nucleotides. This is an example for thymidine. Thymine. This is a thymidylate acid. So based on this, so I can summarize about the nomenclature of these nucleotides. So in case of ribonucleoside and ribonucleotide, in case of RNA, this is an abbreviation for ribonucleoside and tides in RNA. So this is the summary of the RNA, and this is the summary of nucleosides and nucleotides in case of DNA. So this is the overall summary. So if the base present with ribose, in case of RNA or deoxyribose, it is called as nucleoside. If this nucleoside attached to base and nucleotide base and the phosphate group, which gives a structure called nucleotides. So in this base position, any of these nucleotides are present depending upon whether it is RNA or DNA. So this is the summary of the nucleosides and nucleotides. So now we will come to Chargaff's rule. So I told when I was introducing the historical milestones of discovery of DNA. So in 1950, by a scientist called Erwin Chargaff, made an observation on the bases and other components of DNA. So according to his rule, the first rule is purines and pyrimidine bases are in equal amount. That means if you add the adenine and guanine, they are equal. Adenine plus guanine, and is equal to thymine and the cytosine. A plus G is equal to T plus C, or A plus G divided by T plus C will be always equal to 1. So this is the first rule this Chargaff gives. That is, the base pairs are in equal amount. Purine and pyrimidines are in equal amount. And the second one is the molar amount of adenine is always equal to molar amount of thymine. That means you know that adenine pairs with thymine. That means these are like partners. So these are in a same molar amount. Similarly, the molar concentration of guanine is equal to molar concentration of cytosine. So this is because the guanine will always pair with cytosine with 3 hydrogen bonds. So this is the simple rule. Adenine is equals to thymine and guanine equals to cytosine. So if you take the ratio of this adenine to thymine, it is 1, and guanine to cytosine, it is also a 1. And the third rule is sugar deoxyribose and phosphate groups occur in equimolar proportion. The third observation what he made was the number of sugar molecules and the phosphate groups are also occur in equimolar proportions. And the fourth one is adenine thymine base pairs are rarely equal to CNG base pairs. This is very, very important. So AT base pairs like A pairs with T and C pairs with G. So number of these AT pairs and GC pairs may not be equal. And the fifth one is the ratio of A plus T divided by G plus C is variable. This is variable but constant for a species. It can be used to identify the source of DNA. The ratio is low in primitive organisms and high in advanced ones. So that means according to his theory, so A plus T is always A plus T. The ratio of A plus T and G plus C is variable across different organisms, but it is very constant for the particular species. So based on this introduction, so we will discuss about the double helical structure of DNA, which was given by Watson and Crick in 1953. So this was this is very important question. So last time also they asked for 5 marks, and I don't know about this time, but whatever I am telling, I am stressing, these are very important. So Watson and Crick used the chemical analysis and X-ray crystallographic studies to propose double helical model of DNA. This structure became universally accepted soon after the discovery. So this is the structure. There are 2 polynucleotide chains. Just look at this cursor. These are the 2 polynucleotide structures which has sugar, phosphate and nitrogen base in the backbone. So this is called as a major groove and this is called as a minor groove, and one runs in. So I will discuss in the future slide about the salient features of this double helical structure. So these are the hydrogen bonds which are found between guanine and cytosine, and these are the 2 hydrogen bonds found between adenine and thymine. And what are the main features of this double helical structure is DNA molecule is made up of 2 polynucleotide strands, as I told. And the 2 chains of the DNA have anti-parallel polarity. That means one polynucleotide strand runs in 5' 3' direction and another runs in 3' 5' direction. That means these 2 chains run in 2 opposite direction. So backbone of each polynucleotide chain is made up of alternate sugar-phosphate groups and the nitrogen bases project inwardly. So these polynucleotide strands are made up of alternate sugar-phosphate groups. So nitrogen bases of 2 polynucleotide chains forms a complementary pairs. So A opposite T and C opposite G. That means A pairs with T with 2 hydrogen bonds and C pairs with G with 3 hydrogen bonds. So this is about pairing. So how the thymine and adenine pairs with 2 hydrogen bonds, and how guanine and cytosine pairs with 3 hydrogen bonds. So these hydrogen bonds are very weak bonds. So upon heating, you can break these 2 polynucleotide strands. That is called denaturation. That means if you isolate the DNA and heat it at 95 degrees centigrade, the double helical DNA will become single stranded. So because these hydrogen bonds will break, because these are very, very weak in nature. So even if you change the pH of the solution, so these hydrogen bonds can be broken and this double helical structure of DNA can be made into single stranded. So this breaking of double stranded DNA is called as a denaturation. And if you cool it, if you cool the solution and change the pH, so these two polynucleotide strands will rejoin. This is called renaturation. So denaturation and the renaturation. And the sixth feature of this double helical structure is coiled in a helical fashion. So that's why it is called double, means two polynucleotide strand. Helical means these are twisted or turned. The coiling is right-handed. So whatever they told, whatever they proposed is the DNA which has a right-handed coiling pattern. So because of this coiling pattern, this produces major groove and the minor groove and the major groove alternatively. So this is how, like I would have shown in the classroom, like how this left-handed and right-handed coiling can be made using just two fiber cables. So, and the seventh feature of this double helical structure is the diameter of this helical structure is 20 Armstrong unit, and pitch, the length of helix to complete one turn, is 34 Armstrong unit. So if you take one turn, okay, so it measures about 34 Armstrong unit. So you know that on an average, so this turn has 10 base pairs. This turn has 10 base pairs. So 34 divided by 10 is 3.4 Armstrong unit. That means the base interval, or the gap between the first pair of bases, that is 80, and the second base pair could be GC or 80. It could be anything. So from one pair to another pair, so it is 3.4 Armstrong unit. These are all very, very important statistics. So they will ask in MCQ true or false. And the eighth feature of this double helical structure is planes of adjacent base pairs are stacked over one another. This one. The planes of adjacent base pairs are stacked one another along with hydrogen bonding. The stacking confers stability to the helical structure. So because of this stacking, so this gives a stability to the helical structure. So, and the ninth point is DNA is acidic in nature. So you know that DNA is acidic in nature. Now for the coiling or the condensation of this DNA, so this needs histone protein. So we have already discussed this when we are discussing about the chemical composition of chromosomes. So as I already told, the histones are positively charged and DNA is negatively charged. So the histone proteins are positively charged and occupy the major grooves of the DNA at an angle of 30 degrees to helic axis. This is very, very important point. Histones are positively charged. They occupy the major grooves of the DNA. So if there are any errors during replication process, will leads into occurrence of mutations. So we are not discussing about mutations. So we have a separate chapter on mutations in genetics course. We will discuss there. That means if any mistakes made by DNA polymerase during DNA replication, which leads to mutation. So now we will study, so what are all the different conformational changes are there in the DNA. Conformational changes means coiling pattern. Conformation means not conformation, not FIR, it is FOR, conformational changes. So variation in the conformation of the nucleotides of the DNA associated with conformational variants of DNA. That means So there is a different conformations of DNA are available. So far, there are 6 different forms of DNA are existing. That is A to E and Z. That means A, B, C, D, E, and the Z form. And out of these 6 different forms, B, A, and Z forms are very, very important. And B form is the actual double helical structure, which is proposed by Watson and Crick, and which is the predominant one. So out of these 6 different forms. So this question was asked the last exam. So what is the difference between A form and B form of DNA? So A form is called as alternate form of DNA, and B form is called as bent DNA, and Z or Z form of DNA is called as zigzag. So these are all the parameters I have compared across these 3 different forms of DNA. It's about overall proportions and rise per base pair, and the helix packing diameter, and helix rotation sense. See, A forms and B forms are right hand coiling, whereas Z form is left handed. So base pair for helix repeat and base pair for turn of the helix. So so in case of B form it is 10, so whereas in case of A it is 11, whereas in case of Z there are 12 base. So these are the differences between these 3 forms of DNA. So this one, you go through it. It is very simple, and so you write according to the question asked in the exam. So this is about B form. This is right hand coiling, and A form is also right hand coiling, and this is Z DNA, which is a left-handed coiling pattern. So how to measure the size of the DNA molecule? The size of the DNA molecule means how we can measure the length of the DNA. So it is always expressed in terms of base pairs. So why it is pairs? Because there are 2 polynucleotide chains are present. So usually these are expressed in kb, that means kilo bases. That means 1 kb is 1000 base pairs are there. So 1 kb DNA means 1000 base pairs. 1 MB is and 1000 kb are 1 followed by 6 zeros. So 1 million base pairs or 10 lakh base pairs are present in that particular DNA. Or you can also mention it as GB, like giga bases, not gigabytes. Don't say word byte. Okay, it is bases, kilo bases, mega bases, and giga bases. So 1 giga base is 1000 MB. It is 1 followed by 9 zeros. So this is how the size of the DNA is measured